Hello and a very warm welcome to everyone joining us for today's Drug Target Review webinar titled Advancing Obesity Drug Discovery, Cell-Based Assays for GLP-1 and the G-Suite. My name is Dr. Carrie Haslam, Editor at Drug Target Review, and I'll be moderating today's session. I'm delighted to be joined by Dr. Frances Reichert, Technical Specialist for Biologics at Eurofins BioPharma Product Testing, and Shruti Kumar, Field Application Scientist at Eurofins DiscoverX. Our experts will explore the challenges and opportunities in obesity drug discovery, highlighting a range of well-characterized assay solutions that have supported over 100 obesity therapeutic programs worldwide. Our experts will also present their portfolio of pre-qualified functional assays designed to enable effective screening and characterization of therapeutic candidates targeting obesity-related GPCRs. Following the presentation, we'll move on to our question and answer session. So please feel free to submit your questions at any time during the webinar via the questions panel on the right-hand menu. And with that, I'll hand over to Shruti to begin the webinar. So over to you.
Welcome everyone, and thank you for joining this webinar. I am Shruti and I will be guiding you through the first part of our presentation today. We have structured this webinar to provide a focused overview of our qualified cell-based assays and how they can accelerate your obesity drug discovery efforts. Now, take a look at this timeline. It visually tells a powerful story about the evolution of obesity and type 2 diabetes therapeutics. For decades, as we see on the left side of the timeline, the progress was measured, even cautious. While important medications emerged, the overall pace was relatively slow and spaced out for the past 60 years. But something changed fundamentally. Just look at the density of the activity within this recent 4 years, starting from 21 through 2024. We have moved to an era of rapid innovation, which is fueled by a deeper understanding of obesity pathways, particularly those involving GLP-1, GIP, and glucagon receptors, and the power of multi-target drug design. And what's particularly exciting is the breadth of the approaches we are now seeing. Beyond peptide therapeutics, we are also seeing a wide array of small molecule drugs emerging, offering even more diverse and targeted ways to combat obesity. So the buzz around these new obesity drugs like Mounjaro is real, and it begs the question, what's driving their enhanced efficacy and why this shift towards more intricate drug designs? This slide provides a mechanistic answer. The core principle is multi-site of action. Traditional therapies for metabolic diseases often adopted a single target strategy. However, we now understand that metabolic homeostasis is a highly complex multi-organ system. Optimal regulation requires a coordinated action across various tissues as shown here. Now, if you think of agonists as activators, older drugs were often single activators. Now we have unimolecular multi-agonists that are single molecules like tirzepatide and retatrutide that can activate multiple receptors like the GLP-1, GIP, and glucagon, and all at once. This multi-pronged strategy is the secret to their enhanced efficacy. But this increased sophistication in drug mechanism also means we need the right tools to understand and characterize them thoroughly. And that's where our advanced cell-based assays come into play, which we will explore in the upcoming slides. Building on our understanding of multi-site mechanisms, this slide directly addresses how we as scientists can characterize these complex drugs using cell-based assays. The obesity therapeutic pipeline is dominated by GPCRs like GLP-1, GIP, glucagon, and others like amylins and PYY, among others. Throughout the drug development stages, from screening to potency lot release, specific cell-based assays can provide critical information for deeper understanding of the drug's pharmacological profile. It can guide optimization of such profiles and de-risk development programs. Now, given that these GPCRs are the key targets, we have key assay formats like cyclic AMP accumulation, beta-arrestin recruitment, and receptor or internalization, which serve as invaluable investigative tools to evaluate ligand bias, ensure advantageous design, wherein you can have reduced undesired on-target signaling, and together these establish an accurate drug pharmacology profile. And in the later stages, these cell-based assays remain essential. These are vital for generating characterization data for regulatory submissions, including NAP testing to assess immunogenicity and for ensuring consistent product quality through GMP potency and lot release testing before commercial release. Ultimately, cell-based assays are not just an afterthought. They are the core to accelerating the development and ensuring the success of these vital obesity therapeutics at every stage. Now, let's take a quick look under the hood at the technology that powers many of DiscoverX's cell-based assays. It's called enzyme fragment complementation, or EFC. Think of it like this. Imagine a puzzle of an enzyme, which is a beta-galactosidase enzyme, or beta-gal for short. We split this enzyme into two inactive pieces, the enzyme donor and the enzyme acceptor. Neither pieces work on its own. However, when these two fragments are brought together, that's the complementation part. They snap back into a functional active enzyme. And this active enzyme can then act on a substrate to produce a light signal that we can easily measure. Why is this clever puzzle important? Because we can link these enzyme fragments to cellular events. For example, we can attach them to parts of a receptor signaling pathway like the beta-arrestin as shown here. Now, when a drug activates the receptor and triggers beta-arrestin recruitment, it brings the enzyme fragments together, causing complementation of the enzyme fragments and generating a light signal. The beauty of EFC is its flexibility and sensitivity. It provides a robust and quantitative way to measure a wide range of cellular responses, making it a very powerful tool for characterizing drug interactions with its GPCR or other targets. Essentially, EFC technology allows us to see and measure the true MOA of the drug, giving us crucial data for drug discovery and development. This slide showcases our key cell-based assay formats and their diverse applications. We offer a suite of assays specifically designed to interrogate the three key GPCR MOAs, all built upon the robust EFC technology platform. Let's begin with cyclic AMP accumulation assays. This format measures the changes in cyclic AMP levels, which is a universal and a highly sensitive indicator of GPCR activation, providing a homogeneous readout of drug potency. We also provide a beta-arrestin recruitment assay. These assays are crucial for a deeper understanding of biased agonism and receptor pharmacology beyond just the canonical cyclic AMP pathway, giving a more complete picture of a drug's mechanism of action. And for assessing longer-term receptor modulation, we offer receptor internalization assays. These formats help you understand receptor trafficking and desensitization, which are important determinants of long-term drug efficacy and safety profiles. Importantly, all of these assays share key advantages. They are homogeneous, they're sensitive, they're highly robust, and they utilize a simple luminescent readout platform, which makes them really easy to transfer to different labs or CROs, ensuring seamless integration and broad applicability. But these assays are not just for research, they're essential for achieving regulatory approval. Indeed, these assays are specifically designed to define precise mechanism of action, conduct potency testing for GMP and lot release, performed detailed ligand bias studies, facilitate NAP testing, and even confirm biosimilarity, thereby covering a wide range of regulatory requirements. Now, we are moving into a key concept driving the development of next generation therapeutics, biased agonism. This slide illustrates how bias signaling can lead to improved therapeutic outcomes. A traditional unbiased agonist, like shown here, when it binds to a GLP-1 receptor, It equally activates both major signaling pathways. First, it activates the cyclic AMP pathway, which results into desirable effects like insulin secretion, but it also activates the beta-arrestin pathway, which triggers receptor internalization and desensitization. Now, receptor internalization and desensitizations aren't inherently bad, but in chronic therapies, they can become limiting factors. They can result in shortened action duration and potentially reduce long-term insulin secretion. In contrast, with a biased agonist like tirzepatide, the focus is on maximizing CAMP activation while minimizing beta-arrestin signaling. This selective pathway activation leads to key therapeutic advantages, which is reduced receptor desensitization and prolonged receptor occupancy. These advantages directly translate to enhanced and sustained insulin secretion and potentially improved glucose control, along with a better tolerability profiles for patient. Ultimately, by selectively engaging beneficial pathways, you can design next-generation drugs with enhanced efficacy, improved durability of action, and potentially fewer side effects. And as we'll demonstrate further, Eurofins DiscoverX assays are essential tools in the quest to identify, develop, and characterize these biased agonists. Now, let's take a look at some real data that clearly illustrates bias signaling in action. This slide presents data from our cell-based assays directly comparing the signaling profiles of exendin-4, which is a known unbiased GLP-1 agonist, with tirzepatide and retatrutide. In the CAMP accumulation assay, you can see that all three ligands, exendin-4, tirzepatide, and retatrutide, indicate a potent CAMP activation. In fact, tirzepatide and retatrutide show comparable maximum CAMP response compared to exendin. This confirms their strong agonist activity at the CAMP pathway. But this picture changes dramatically in the beta-arrestin readout. Curves for tirzepatide and retatrutide are shifted significantly to the right, indicating they are much less potent at recruiting beta-arrestin compared to exendin-4. Given that receptor internalization is downstream of beta-arrestin recruitment, the receptor internalization assay further supports these findings. As expected, tirzepatide and retatrutide also exhibit markedly reduced receptor internalization capacity compared to the unbiased agonist. This is a direct experimental evidence of bias signaling demonstrated using our cell-based assays. And as we discuss, this bias profile is strongly linked to the improved therapeutic properties observed with these next-generation obesity drugs. Now, I want to showcase a truly innovative assay that takes our cell-based assay capabilities to a step further, the inducible CAMP Hunter GLP-1 receptor assay. What's unique about this assay? As the title suggests, it's inducible. We have engineered our GLP-1 cell line where the expression of the receptor itself is controllable using doxycycline or DOX. This is a game changer because for the first time, we can directly study the impact of receptor density on drug potency and efficacy, all within a single, highly controlled assay system. These graphs showcase this beautifully. For each ligand, exendin-4, tirzepatide, and the small molecule GLP-1 agonist orforglipron, we tested responses at increasing doses of doxycycline. You can see that as we increase the dox, signal intensity increases dramatically, and the dose response curve shifts, demonstrating that as we increase the receptor expression, the apparent potency of these agonists also increases. This provides a deeper, more nuanced understanding of GPCR signaling and drug action and is a powerful tool for advanced drug discovery and research. So far, we have covered the why and the what. Now let's move to the how. This case study will walk you through the actual process of bioassay development, showing you how we design, optimize, and pre-qualify these assays to ensure they are robust and fit for purpose. At the heart of our strategy are industry requirements. We understand that you need assays that are not just scientifically sound, but are also practically useful and regulatory compliant. This is why our assays are MOA reflective, their specificity and stability indicating, and are repeatable, robust, precise, and accurate. To achieve this, our assay development process is meticulously designed. Some of the key parameters include cell density titration, optimizing the ligand dilution schemes, and optimizing the detection reagent and read time to maximize the signal quality and assay performance. And our assay pre-qualification follows the ICH guidelines, which means we systematically evaluate key assay performance parameters shown here. So together, we also provide long-term support. This includes maintaining dedicated and well-characterized banks to ensure consistency in the assay performance over time, establishing supply agreements for risk mitigation to guarantee reagent availability and ensure full traceability. Our 15 plus years of experience speaks to our track record and our deep understanding of bioassay development and qualification. More on the robustness of our assays, this slide visually and statistically demonstrates the plate uniformity of our GPCR bioassay. What we are looking for is consistent performance across the entire plate. The key metric to focus here is the percent CV across the plate. This value, just at 8%, demonstrates the remarkable low variability across all wells of the 96 well plate. Now, looking on to our pre-qualification study design. To start with, we rigorously assess repeatability. As you see in the table, over 6 distinct days by a single analyst at the 100% nominal concentration. For relative potency estimation, we challenge the assay across a broad concentration range from 50% to 200% of the nominal, ensuring it accurately measures potency across that spectrum. Finally, to confirm intermediate precision, we simulate real-world lab conditions by incorporating multiple analysts, testing over multiple days, utilizing 4 different nominal concentrations, and even introducing 2 distinct lots of our bioassay cells. And our predetermined success criteria is set at 100% success rate, meaning absolutely no failed plates for both system and sample suitability. Following the relative potency design we outlined, this slide presents the actual results. And as you can see, they are exactly as expected. The consistent curves across the analyst and concentrations and the close potency values reinforce the assay's accuracy and reliability in measuring relative potency. All right, now that we have explored the rigorous study design and the key data on potency estimation, let's turn our attention to the overall summary of our GPCR bioassay pre-qualification, just captured on this slide. Focusing on the first data table here, we see the results of our accuracy assessment. We tested the bioassays at various expected potency levels ranging from 50 to 200%. This is a range that is relevant to real-world sample testing. Recovery percentage tells us how well the bioassay recovers the true potency. Ideally, we want to see recovery values consistently close to 100%. And as you can see here, across all the tested potency levels, the individual recovery values hover very close to that 100% mark. Looking at the linearity graph on the right, we are assessing that the assay response is directly proportional to the drug concentration. The R-squared value, which is a measure of linearity, is at 0.97. And R-squared this close to 1, as you may know, signifies excellent dilution linearity. This assay demonstrates a highly linear response, which is essential for your accurate potency determination. Finally, at the bottom, the table summarizes this key parameter data, comparing the actual achieved values against our predefined specifications. Similarly, for the GLP-1 bioassay, the pre-qualification with tirzepatide data speaks for itself. Across accuracy, repeatability, intermediate precision, and linearity, both these assays for tirzepatide have consistently met and in some cases exceeded our stringent pre-qualification criteria. This comprehensive pre-qualification data definitively proves that the assay is reliable and will be a high-performing assay in your labs. Now, to broaden our perspective, let's take a step back and look at the bigger picture, the entire drug discovery process. This slide illustrates how critical it is to have a phase-appropriate format of cell-based assay at each stage of drug development. In the early screening phase, you need assays that are geared towards hit identification, often requiring high throughput capabilities. As you progress to characterization, the focus shifts to understanding the pharmacology, requiring multiple MOA-based assays. And then as you move closer to commercialization, potency testing and commercial lot release become paramount, demanding a highly robust, reproducible, and a qualified assay for lot-to-lot consistency and regulatory compliance. We understand this phase-dependent need intimately, and we offer phase-appropriate solutions tailored for each of these critical stages. For early stage screening and characterization, our stable cell lines are ideal. They are MOA reflective and importantly offer very high passage stability. This ensures consistency and reliability for those early large-scale screening campaigns. As you need to scale up, our high-density cell banks become invaluable. They are scalable from 96 well all the way to 1536 well formats. As for the later stages, particularly the potency testing and commercial lot release stage, our thaw and use pre-qualified bioassays are the perfect fit. As we have seen in the previous slides, they offer a high reproducibility, which is absolutely crucial for lot-to-lot consistency. And importantly, they are very easy to use, streamlining your QC processes and reducing complexity. Now, here is a snapshot of our extensive portfolio. As you can see, we cover a wide range of key targets in this obesity space, starting from GLP-1 and GLP-2R, which are your essential incretin therapies, to GCGR, GIPR, amylins, apelins, GPR75, and many more. Whether you're developing monoagonists, unimolecular coagonist, or any other novel mechanisms, our portfolio has you covered. Critically, for each receptor, we offer a range of MOA reflective assay formats. This allows you to comprehensively characterize your compounds across biologically relevant MOAs, giving you a richer and a more complete pharmacological profile. This comprehensive portfolio empowers you at every stage of drug discovery. We provide the right assays for the right targets at the right phases, all to expedite your discovery and development and for you to advance confidently. Now, as I've showcased the capabilities of our bioassays, a fundamental question you might be asking is, why should you trust DiscoverX assays? It's a fair question. Well, the proof, as they say, is in the pudding. And for DiscoverX assays, that pudding is the industry acceptance. DiscoverX assays are consistently and widely used, cited, and trusted by leading pharmaceutical companies, regulatory agencies, and in cutting-edge scientific research across the globe. So in summary, DiscoverX is the global leader in cell-based assays for obesity and type 2 diabetes therapeutics. We accelerate therapeutic drug discovery by providing assays that are comprehensively addressing multiple therapeutic modalities, from small molecules and innovator biologics to biosimilars and generics. Our MOA reflective assays targeting the key receptors that we have discussed are offered in phase appropriate formats, ensuring relevance throughout your drug development journey. This is why we are highly industry accepted, evidenced by over 50 publications, patents, and regulatory filings from global pharmaceutical leaders in this field. We are already supporting over 25 biologics and biosimilar programs globally, a testament to our scale and expertise. Our assays are pre-qualified with leading obesity therapeutics approved on the market, including Ozempic, tirzepatide, and in clinical trials like retatrutide. Finally, to support your path to commercial drug release, we offer ICH-based pre-qualified bioassays, custom assay qualification services, global method transfer support, and a long-term support commitment, ensuring your success from development through commercialization. Thank you everyone for your attention, and I will pass on to the next presenter.
Thank you for the introduction. It's a pleasure to have the opportunity to share our recent advancements in semi-automated potency assays and discuss how automation can improve both the consistency and efficiency of potency assays. I will start with a short introduction to Eurofins and potency assays. Subsequently, I will discuss the key challenges of bioassays and walk through a case study that highlights our semi-automation approach. Finally, I will summarize our findings and share key takeaways for you. Now, before diving into the technical details, let me give you a brief overview of Eurofins. Eurofins is a global leader in laboratory services offering testing solutions for industries such as food, environment, and chemicals. Additionally, our Eurofins BioPharma Product Testing division is specialized in providing comprehensive testing services for the pharmaceutical industries. Eurofins BPT is the world's largest network of harmonized GMP testing laboratories with 40 locations across 20 countries and a team of over 8,000 employees. At Eurofins BPT, we support the development and manufacturing of biopharmaceuticals, starting with raw material testing required for product manufacturing until quality control testing for final product and release for the market. We have extensive knowledge in biologics, such as monoclonal antibodies, peptides, proteins, vaccines, but also new modalities like cell and gene therapy and different types of oligonucleotides. We offer a full range of biopharmaceutical testing services, including safety, physical chemistry, identity, purity, impurity, but also cover stability studies as well as commercial release testing. Today, we will focus on potency testing, a critical quality control measure for biologics. Potency assays are essential for biologics as they are used to measure the biological activity or potency of biopharmaceuticals. They are required as quality control tests for all biologics according to ICH guideline Q6B. Regulatory agencies like FDA and EMA emphasize that potency assays have to align with the mode of action of the biopharmaceutical to provide physiologically relevant results. Essentially, bioassays are a holistic quality control tool ensuring that each drug as well as each manufactured batch performs as expected in patients. Bioassays present significant challenges as they are very complex due to the use of living cells, multiple pipetting steps, handling of small volumes across one or more 96 well plates. A simple method, as shown here, can involve seeding of the cells in a 96 well plate, preparing dilutions and serial dilutions in one or more 96 wells, and mixing them all together. During this incubation period, the biopharmaceutical has the same effect on the cells as it would do in any patient. For instance, a monoclonal antibody killing a cancer cell. Addition of substrate allows the detection of the drug's response, which is finally measured with an appropriate instrument. The number of these volume transfers can easily involve 100 pipetting steps, which introduces viability and lowers reproducibility when performed by different analysts or else on different days. To address this, labs traditionally use replication strategies, such as increasing the sample replicate numbers or the number of 96 well plates, but this lowers sample throughput and makes scaling difficult. Furthermore, the significant number of pipetting steps also requires extensive analyst training, increases the likelihood of human error, and can even lead to health problems for lab analysts. Automation has emerged as a powerful tool to address these challenges. Full end-to-end automation, meaning from sample receipt to final COA release, remains challenging in a GMP environment. So we focused on semi-automation in a modular manner. Since we can break down bioassays into smaller steps, this approach allows us to automate only certain parts of the assay. This procedure offers high flexibilities in terms of the used instruments and also can account for different method procedures. For highly sophisticated robots, we do require also highly skilled subject matter experts. However, for less complex systems, they are designed for ease of use. So basically, the analyst just press a button in order to start the program. We also can reduce the effort for analyst training and increase our sample throughput. The automated bioassays also benefit by an improved transferability in terms of lab-to-lab transfers as well as analyst-to-analyst transfers. After market research, we selected at first the vial flow 96 from Integra. This pipette consists of 96 individual channels and is used to transfer the volumes of 96 wells in one step. In contrast, our next system we acquired was the Integra Assist Plus, representing a small pipetting robot, and allows the automation of more complex pipetting steps like serial dilutions. This system comes with the ViaLab software to program the pipetting workflow. Both instruments can be placed in a laminar airflow for aseptic working conditions and are applied in GMP sample testings in our labs already. We implemented an even more advanced system. The Hamilton Microlab Star has been set up with a 12 single-channel pipette system, HEPA hood, plate shakers, and a heating and cooling unit. We fully integrated an ELISA plate washer and a reader for data measurement, so this system can run an experiment almost on its own. Each of these platforms provides varying levels of automation and flexibility and comes with their own pros and cons. In our case study, we evaluated the potency of a biopharmaceutical peptide using the CMP Hunter bioassay kit of Eurofins DiscoverX. All components needed to perform the assays are included in the kit. CHO cells are seeded in a 96 well plate and are placed in a 37-degree incubator. On the next day, agonist dilutions are prepared, transferred to the assay plate containing the seeded CHO cells and incubated together. After adding all the required detection reagents, we measure a response signal, and by plotting this measured values against the agonist concentration, we can generate a sigmoidal dose response curve as shown here in an example. Initially, we performed the assay manually using conventional 12-channel electronic pipettes. We used the agonist as reference standard at 100% activity and at low and high potency levels, 50% and 200%, to assess accuracy and precision of the assay at the range of the method. In this figure, we plotted on the y-axis the measured luminescence values and on the x-axis agonist concentration. The reference standard curve is depicted in red, whereas the sample curve is shown in green. As a measure of our results, we divide the measured potency values with the expected potency values to calculate the recovery. And in an optimal result case, we get 100% recovery. The results shown here for 50% expected potency value, we have a recovery of 114%, which is slightly overestimated. On the other curve, you see that we get for the 200% nominal potency higher replicate variability. We then use the Integra Assist Plus to semi-automate the entire assay performance, starting from cell seeding to final substrate addition, highlighted here in blue. The same reference standard concentration and sample concentration were used as for the manual assay. However, these results showed not only a strong overestimated potency for both levels indicated by the high recovery values, but also an increased replicate variability and a significant dose response curve shift. This suggested that further optimization was needed. For root cause investigation, we re-evaluated all programming steps and pipetting steps. we identified a too slow addition of the detection reagents as the most likely root cause. To address this, we tested two approaches shown here. For option 1, 2 steps were automated, shown in blue, so meaning serial dilutions and dilution transfer. Whereas for option 2, we automated again 4 steps as before, but with lower pipetting speeds. So for both options, we improved the pipetting modes and additionally switched from the Integra Assist to the ViaFlow 96 for increased efficiency for the dilution transfer step. Now here I'm showing a short video demonstration of the Integra Assist system in action. So first, it can be used to dispense media in deep well plates or any kind of other plates or even Eppendorf tubes. The pipette is also able to perform serial dilutions on their own using different volumes for each transfer as well as mixing step. Finally, the pipette can be also used for any kind of volume transfers. Here we transfer our serial dilutions from the dilution plate in the middle to a wide assay plate. In addition, we applied also a slanted plate holder, which allows tilting of the plate. After optimization, our results become much more consistent, as shown here. On the left side, you see the results with the 2-step automation, and on the right, the results with the 4-step automation. For both optimized methods, the recoveries were with 103% and 96% close to the expected 100% recovery. When testing the method range, for 50 to 200% potency values, we could confirm our previous result. An expected recovery of 100% was measured for 50%, 100%, and 200% nominal potency. Additionally, the results clearly showed a low replicate variability with a dose response curve as with manual performance. So this demonstrated that fine-tuning automation settings is key for reliable results. In our case study, we semi-automated the potency assay of an agonist using the Eurofins DiscoverX EAMP Hunter bioassay kit. Initial results with the Integra Assist Plus caused the dose response curve shift, increased variability, and unexpected accuracy values. By re-evaluating the programming steps and adjusting pipetting times, we improved precision and recovery to a more accurate level. This case study taught us several important lessons. So first of all, automation alone isn't enough. So fine-tuning of the pipetting steps is critical. Secondly, semi-automated assays improve assay consistency compared to manual methods. And thirdly, hands-on time can be significantly reduced, allowing analysts to focus on other critical tasks and also reducing the human error rate. Although automation enhances efficiency, careful optimization is the key to success. I would like to extend my gratitude to the whole bioassay automation team, Sean, Sabine, Tabea, Philip, Lara, and Alex. Thank you all for your time and attention today. Please find here our high-level overview. Shruti talked about pre-qualified functional and mode of action reflected cell-based assays, and I talked about semi-automated potency assays. If you need more information, please visit our website shown here. Thank you very much for your attention. We can now answer any questions you may have.
Thank you, Shruti and Frances, for that fantastic presentation. Now let's move on to the final part of today's webinar, which is the question and answer session. So as a quick reminder to our audience, you can still submit your questions using the questions panel in the menu on the right. So let's get started. So the first question we've got is, what are some limitations of the current semi-automated approach and how can they be addressed in future developments? This is one for Frances.
Frances. Yeah, thank you for your question. So basically, each of the platforms comes with their own advantages and disadvantages. The initial setup and qualification or validation of an instrument can be quite cost and resource intensive, depending on the system, and also can require a certain knowledge to get it implemented. So less complex systems like the Integra systems I showed here can be really fast implemented in the labs, meaning within a couple of months. And the operation is very similar to manual pipettes. Also, the Biologics program, which comes from the Integra Assist, is very easy by a drag and drop program. However, when we have to implement the Hamilton system, which is really very complex, also requires an TC for operation, we need more knowledge about the system and also much more time for implementation. So initial setup and qualification is one limitation. Also, ongoing costs should be assessed. For example, with the Wireflow 96, we need 96 pipette tips for the transfers. And depending on the number of plates we have to transfer, this might sum up to a very large number of pipette tips. And this might be then better to switch to another system. And
That's
also
great.
semi-automation, we still require human intervention in terms, for example, of plate handling, reagent additions, and we are also aiming for the Hamilton system, which is of course more advanced, but this requires less intervention by any human analyst. And also, last but not least, there might be also parts of an assay or even assays which might not be possible to semi-automate. So at the end, so for future developments, we really have to check for the pros and cons of each system and have to balance the systems when you're selecting them for the lab work, as well as specifically for your method. And we're also trying to standardize automation protocols for both the systems. Yeah,
That's great. Thank you, Frances. So the next question we've got here is, how would you decide which of these assays, CAMP, beta-arrestin, and receptor internalization, to prioritize in early screening? And are there specific scenarios or target characteristics that would make one assay format more informative than the other?
Yeah, I can take that question. And it's a great question. Thank you for asking. And it's really a central question to effectively, screening your novel obesity therapies. So we'd recommend a strategic approach and usually start with your cyclic AMP assay. It's crucial for identifying agonists that activate like primarily G protein coupled pathways for GPCRs like GLP-1 and GPR, which is also linked to your insulin secretion and metabolic regulation. But following up, we would strongly emphasize on the beta-arrestin assay as well, which is really crucial for characterizing your ligand bias, which has become quite popular with tirzepatide and is suspected to contribute to tirzepatide's efficacy. And finally, perhaps later in the early screening process, the receptor internalization assay could provide a very valuable information on how a ligand accepts receptor trafficking and also impacts the duration of action of your therapeutic drug, which could kind of give you information on the recycling and could be also beneficial for long-acting therapies. But also, again, this question can shift based on your target product profile. So if you're aiming for a maximal insulin secretion type of therapeutic, CAMP might be your first assay of choice. But if you're looking at reducing potential side effects, which is linked to your beta-arrestin pathway, this assay can become really, really important. And if you're looking for sustained efficacy and the receptor trafficking, then the receptor internalization assay would be your choice.
Oh, that's great. Thank you, Shruti. So the next one here is, how do automated systems handle variability in living cell-based assays, and what strategies help mitigate inconsistencies?
I can take this.
Yeah.
So for especially for cell-based bioassay, the question always is how we handle the cell seeding and cell viability, since it's a living system. But we have used all the systems also especially for cell seeding only. So we were also interested how the systems seed cells. And when we seeded 96 well plates and compared to manual performance, we really got the same CVs as with the manual performance. So in our hand, all the systems, including Hamilton, also both the Integra systems behaved quite comparable. And it's really important also to do the programming of the pipettes accordingly. So this means definition of the volumes, the mixing speeds, immersion depths. So in that way, you can overcome any hurdles you might have or you might face in cell-based assays. So it's really important also to have the pipetting modes set also to prevent cell disruption or like that. So in that way, you have to you can overcome the hurdles. So in our hand, it's working quite well.
That's great. Thank you, Frances. This next question is, with reducible cell lines, why does DOX concentration affect potency?
Yeah, another great question. Thank you for asking. So basically what we have in inducible cell lines is that we are controlling receptor expression with doxycycline concentration. Increased doxycycline will lead to higher receptor expression, and in assays like cyclic AMP, this impacts potency because these assays are amplified systems, right? Where single activated receptor can interact with and activate multiple G proteins, each of which can then activate downstream effectors like adenyl cyclase, which leads to a substantial increase in cyclic AMP levels. Higher receptor density would result in a very leftward shift of the dose response, which will showcase a very low EC50, potency. And could lead to an overestimation. So basically, the bottom line is like more doxycycline concentration leads to more receptors, which makes the drug appear more potent because it needs to occupy fewer receptors to achieve the same effect, which is where you can see the potency shift with the receptor density.
That's perfect. Thank you very much. So this next one is, are these assays HTS, so high throughput screening, compatible?
Yes, I'll take that question again. Yes, absolutely. Specifically, if you see the cell line model that we offer is designed for high throughput workflow because we provide a very detailed protocol with a 384 well plate, but these assays can be readily miniaturized to a 1536 well format as well, and we've done that before. So basically, what you're doing is you're enhancing your throughput and reducing your reagent consumption as well. And this scalability would make it ideal for large scale compound library screening and early drug discovery applications.
Perfect. Thank you very much. So this one is, can semi-automation be easily integrated into existing lab workflows, or does it require significant infrastructure changes?
I can take that. So
Yeah.
for semi-automation, it's easy to integrate when you have these small benchtop automation systems like Integra Wireflow and Assist Plus. So as they fit easily into the standard lab spaces, and also most importantly, which was also one important decision point for us, under the laminar airflow, which is needed for cell-based assays. So in that way, they can be easily integrated, which is a little bit more difficult for complex systems as the Hamilton system, as they are much bigger and also operated by a PC, which requires a special software and also validation of this. So infrastructure changes might be the space, so that we have a very low footprint for the Integra systems, but for Hamilton they are much larger. We do need, of course, training requirements, but here it's the same for Integra systems, as they are quite comparable to manual pipettes. From an operational point of view, it's easy to integrate, which is a little bit more difficult compared for Hamilton systems in terms of operation and also setting up new methods. And the best approach for integration, also to get to know for semi-automation, is to start with low complex systems like benchtop systems, and then move over to more complex systems.
Perfect.
So that's the way we propose.
Great, thank you. So we've got time for some more questions, and this one that's just come through is, what are the regulatory considerations when implementing semi-automated potency assays in a GMP environment?
So for the Integra systems, as I said, we handled them as a manual pipette or as a pipette system. So we classified them as a B system, which is according USP 1057. And then for Hamilton system, which is operated by a computer, we classified them as a C system. So this also defines the requirements when it comes to implementation into a GMP environment. So a full validation, including audit trail check, software validation is needed for the Hamilton system, which is not required for the B system. And also we do require the GMP documentations like standard operation procedures, SOPs, and also we follow change control processes whenever there are any changes to the systems. And for pipetting accuracy and precision, we follow the international guideline ISO 8655 in order to prove and confirm that the pipetting the correct volumes. So these are the requirements for the semi-automated systems.
Perfect, thank you. So we've got another question that's come through, and this is, can you compare the pharmacology between the GLP-1R and GIPR cell lines if expression levels different? And I've got in brackets here, have you normalized receptor density across receptors? Not sure who would like to take that one.
Yeah, I can take that one.
Of course.
Yeah, another excellent question. And so the point of comparing the pharmacology between the two different cell lines would be, I'm guessing, to identify if the ligand or the therapeutic under discovery is a full agonist or partial agonist at these receptors. And what we have done is not normalize receptor density across the receptors, but have a control in the assay. For example, we could normalize it to the percent cyclic AMP response, or add a control like a force colon that would maximize, give you a maximal camp response, and then have the data for each of the receptors for the particular ligand normalized to that maximal response by force colon. So there are different ways to go about comparing it, but we have not normalized receptor density across receptors.
That's great. Thank you, Shruti. So we've got another one that's come through, and this is, can the cell-based assays be used for fragment screening? And what is the weakest potency that can be detected?
Again, another great question, but this would typically depend on the ligand that is under consideration or under, but I would say we have cyclic AMP potency readings for multiple ligands in picomolar and nanomolar range. So we have identified, or our assays are able to identify very weak or very like very strong potent agonist as well as weak agonist, but it would be a property of the ligand.
Perfect, thank you. Well, that's all we've got time for today. So first, a huge thank you to Dr. Frances Reichert and Shruti Kumar for a fantastic presentation and discussion. And thank you to everyone joining us online and for submitting your questions. A quick note before you go, as you leave the webinar, a short survey will appear on your screen asking you to rate your experience. If you're unable to complete it now, don't worry, it will be also sent to you via email shortly, and we would greatly appreciate your feedback. And this webinar will also be available to watch on demand in just a few days' time. So on behalf of Drug Target Review, thank you once again to our speakers and to all of you for attending. We hope to see you again soon. Thank you once again for joining us.